Okay, the next modeling we're going to showcase is protein synthesis, and we're going to make this in 2 steps, transcription and then translation. And we know that protein synthesis is the process of making a protein, and of course that all starts with the DNA. So here's your DNA again, 2 strands joined together. We see those base pairs just like we did in the DNA replication video. And of course, because this is DNA, this is happening in the nucleus because the DNA cannot leave the nucleus. And the first step of making a protein is transcription. So transcription of DNA into RNA. And that is the essential that you need to know for this step. So we're going to start with DNA, and this is going to happen in the nucleus. And the RNA polymerase, which is very similar to that DNA polymerase that we saw in DNA replication, the RNA polymerase is going to be the spot on the DNA where the RNA is formed. It's basically going to add those RNA nucleotides just like we saw the DNA polymerase do. It's very similar. And so the DNA has to open up or unzip, and then the RNA polymerase is going to add based on the sequence of the DNA. So that very first nucleotide that I see is a guanine, and so I'm going to know that the RNA polymerase is only going to allow a cytosine to add on. Now I do want to show you this key that we have that shows that there are some similarities between the DNA and RNA nucleotides. They look very similar. 3 of the colors are the same. That's because adenine, guanine, and cytosine are all going to be both the same on DNA and RNA. And then for RNA, we're going to notice that we have a uracil, which is shown in white, instead of thymine. But the base pairing is going to be the same. Just substitute a uracil for a thymine. So the other thing to notice here is that the shapes of the RNA are a little bit different. That's to showcase that these are RNA nucleotides. They have a ribose sugar instead of a deoxyribose sugar. But they do look very similar. So I'm also going to make sure that this is going in that anti-parallel fashion, and that's going to join here. And next would be adenine because I have a thymine right here, so it's going to add. And this RNA, these are going to join together, but it's really not going to hook on. This RNA is going to make its own strand. We're just kind of using the DNA for a template. And as transcription occurs and moves on, we see the RNA strand starting to form, and it is the growing RNA strand, and it kind of goes off to the side. And then as the DNA continues to be used, it does kind of come back together into its original format. So it is basically unharmed in this process, and we're going to keep this going until we finish this section of DNA. I do want to point out that this process is only to make a protein, so we are just going to be utilizing one section of DNA, a section called a gene. And so depending on what protein is being made, that's the section of DNA that's going to be used as a template. Okay, once the process is complete, there's the DNA, has been unharmed in this process, and then here is my messenger RNA. That is the product of the first step of transcription, and that messenger RNA is going to leave the nucleus through the nuclear membrane, through a pore, and it is going to go out into the cytoplasm and get ready for the next step, which will be translation. Okay, before we move on to translation, which is the next step, I just want to showcase these tRNA molecules. The tRNA molecules each have 1 amino acid, and they're showcased in this modeling with some different colored stickers to show that each tRNA is bringing a different amino acid. There are 20 possible amino acids, and which one is attached to the tRNA has to do with these letters at the bottom, the ACC, the anticodon. And of course, we're going to see which one gets brought based on the mRNA sequence. So there's our messenger RNA. It has left the nucleus and gone out into the cytoplasm and has found a ribosome. And now we're ready for translation. This is where RNA is going to be used to create a protein. So the messenger RNA basically feeds through the ribosome, kind of like a little conveyor belt, if you will. And the tRNA that is going to arrive is going to match up perfectly with the mRNA, and that is not a coincidence. And so the first one here that is brought is called our start codon. and it is MET. Anytime we read a section of RNA, we're always going to start with the codon AUG. And that is just the start codon, and that basically signals, hey, this is the part of messenger RNA we want to translate. It's time to get started. So if you notice then on the messenger RNA, the next is ACC. So I'm going to look for a tRNA that has the complementary base pairs in its anticodon. And so there it is, and it's going to join together. And when that happens, this whole thing kind of gets scooched over. And this amino acid basically hooks onto the next tRNA. It hooks on here, and then this tRNA is free to go. Okay, and it may be reused, it may not, but it is free to go. It's going to go get another amino acid. And then this is going to continue. So next is GGU. So I'm going to look for the one that fits right there. And there's only one that's going to fit. And it's carrying a certain amino acid. And then again, we're going to see these amino acids join together as the process continues. And here we show the process having almost completed. And so here we have our long amino acid chain, which once it gets to a certain length, then we call it a polypeptide chain. And then eventually a little longer, we call it a protein. And this last tRNA is going to drop off its amino acid. It's going to add to the growing chain. Then you'll notice there is a stop codon. There are three stop codons, sections of RNA that code for it's time for this process to be over. And so then this RNA is going to be released, the tRNAs are going to be released as well, and then the protein is what's left. Now, this is a simplification. This protein would be much longer and would eventually fold into its functional shape and then go off and do whatever its protein job is to do. So this process is the same for all proteins. They follow these same exact steps. And this order of the amino acids, which is originally from the order of the DNA sequence, determines what kind of protein this is. So again, we have 20 different amino acids, but the sequence that they are in is going to determine what kind of protein has been made.